I noticed that photosynthetic bacteria is in the trend for a while. If you don't know, photosynthetic bacteria, or PSB, are anaerobic bacteria that perform photosynthesis. Believed to be one of the earliest life forms on Earth, photosynthetic bacteria actually build the world that we live in today. It has been found pretty useful in agriculture, aquaculture, waste treatment, and other fields, and that's why it's being widely used right now. Traditionally, PSB can be categorized into two groups: the oxygenic, which are able to produce oxygen, and anoxygenic, which are not. From the figure, you can see these two groups. The oxygenic groups, including cyanobacteria, which most of you might already heard of, and the anoxygenic group can be further divided into two, based on what they use as electron donor when conducting photosynthesis. At the end, we have four groups of bacteria: the purple sulfur bacteria, green sulfur bacteria, purple non-sulfur bacteria, and green non-sulfur bacteria. Each group consists of many different species of bacteria with their unique characteristics. Note that new photosynthetic bacteria are still being discovered, so this classification is not definitive. So where can we find those bacteria? Photosynthetic bacteria can be easily found in aquatic environment in low amount. In fact, some of them are so resilient you can find them in almost any wet place. So now I decided to find some myself. There are a lot of videos about how to do it. One popular method is to use 1 egg, 7.5 grams of fish sauce or soy sauce, and 7.5 grams of MSG as growth medium, and mix with water collected from the sample site, usually from lake or river. Put the mixture under sunlight and you will get the bacteria in about a month. So I tried this method with the water I get from a stream and fish pond. The result is not good. All I get is black, stinky liquid. I assume the sugar in my soy sauce makes some other bacteria overgrow. So the second time I try a more refined medium with mostly inorganic ingredients, which does not yield any meaningful result. And that's the time I think maybe the bacteria in my sample is just too few to start with, so I really have to bring out the big gun. A century-old method that allows two people to culture a large diversity of microorganisms is called the Winogradsky column, invented in the 1880s by scientist Sergei Winogradsky. It is easy and simple to make. Here I have some eggshells as a calcium source. I mix them with paper to provide cellulose. Give it a nice crush, then mix it with some egg yolk. I'm using cooked egg in the video, but I later find out you can use raw egg as well. Then I mix those ingredients with the mud I collect from a river shore, and put the mixed mud into a tall bottle about 1/3 of its height. Next, I put more mud into the bottle for around another 1/3 of it. Finally, I fill the bottle with the water I collected from the river and put the lid on. I left the bottle on a grass field which it can be exposed to sunlight, and I made the second bottle with a sample taken from another sample site. 1 month later, the column became fascinating and beautiful with colors. The colors you see are different group of photosynthetic bacteria that grows in different layers of the column due to the different concentration of oxygen and hydrogen sulfide. I take some of the bacteria from this red layer out and put it into the egg medium, and I successfully grow a bottle of purple sulfur bacteria. Now let's just look at the two columns I made. I plan to dig more into these methods in the future. The huge column I use is not easy to handle or extract bacteria from, so I want to find an easy way to do it. Also, I hope I'll be able to isolate some of the bacteria from the column, but that will require anaerobic culture equipment, so you will have to wait. And this is for today's video. Thank you for watching. I will see you next time. Bye bye.